Let's say we've identified an acceptable target. This target has been studied extensively and is thought to play a key role in the progression of disease. The process is now handed off to the second stage to screen this compound for hits that bind to the target. This approach is responsible for the discovery of the majority of today's marketed drugs and is called a target-based screening approach. Often at this stage, a scientist may have an idea of the class of compound that might or might not bind well with the target. Using this information, the number of compounds for initial screening can be reduced, allowing for a more informed approach. Additional reduction of the number of compounds to be screened in the lab can be accomplished using computer software and virtual compounding libraries. When lab screening does commence, it is often accomplished in multi-well plates containing the targets of interest and uses automated equipment to allow for higher throughput. This automation allows for very large libraries of potential compounds to be evaluated in a relatively short amount of time. The target-based approach to screening has proven very useful. However, some scientists believe that its limiting criteria might prevent novel or unexpected discoveries. Target-based screening assumes that we know definitively how that target affects disease, and that the better a chemical binds to that target, the better the outcome we could expect. This is not always the case. Diseases can be very complex, and at times, understanding every mechanism of disease progression and every role drug targets play in that disease is nearly impossible. With this in mind, drug developers have begun to use what is called a phenotypic approach to drug discovery and screening as an alternative to the target-based approach. In a phenotypic screen, a scientist has a living model of disease to test for hits. However, the target and the mechanism of action remains unknown to the scientist. The only thing the scientist investigates is if the compound has been able to somehow reduce or eliminate the signs of that disease. Often, these living models are cells, tissue cultures, or mice that have been developed to model a human disease. The benefit of this approach is that potential for first-in-class compounds is much higher than in the hypothesis-driven target approach. Also, since the target is unknown, further chemical modifications to a hit compound can be very difficult in the phenotypic approach because it is unknown how this change might affect binding to the target. Now, we should also mention here that there is a more modern class of compounds that does not use either of these two screening pathways. Compounds referred to as biologics are produced from a living system rather than in a chemistry lab. These compounds also behave differently therapeutically. Rather than binding to a very specific target, some biologics just copy an existing system in the body. An example of this is insulin, which is a naturally occurring substance secreted by the pancreas but can be administered to diabetics to help regulate blood glucose. For use as a therapeutic in diabetic patients, insulin was originally isolated from the pancreas of cows and pigs. However, it can now be produced with genetic engineering by inserting the human insulin gene into bacterial DNA. As these bacterial colonies divide and grow, they read the inserted human gene and produce insulin. Newer biologics actually do have a more targeted approach. However, they still mimic the body's own targeting mechanisms to take action against disease. Antibodies and proteins can be tailor-made to bind with disease-causing cells or interfere with chemical signaling that progresses disease.